Thank you very much for inviting us today. I am Xavier Heilingenstein. I'm the Chief Scientific Officer of CryoCapCell, a small company specialized in correlative light and electron microscopy technologies with a special focus on high pressure freezing. And Miriam.
Hello, I'm Miriam Lucas. I'm staff scientist at ETH Zurich at CLEM. So that's a microscopy facility there. And I've been in the field of correlative microscopy for nearly 20 years by now. Although I haven't done so much in-resin fluorescence so far, but this is where Xavier comes in.
So what we would like to touch today is about our most recent, our review we did 2 years ago on in-resin fluorescence preservation and what it could bring to your science and how to deal with it. But the thing is, in-resin fluorescence, everyone knows that it's a very great technique, but it's not that easy to achieve. And so we'll come over some of the technical aspects of this to make it work. But in-resin fluorescence is one specific topic out of correlative microscopy, and correlative microscopy exists in so many different ways. It was one of the first use of electron microscopy when David and Sharp in 1944 had to prepare the sample in light microscopy to confirm that their electron microscopy preparation was good. But then if we look at publications on PubMed, we realize that it has been around for already quite some time, but with a significant acceleration since 2003 around, where we started to have more and more of this use and this technological development associated. But before 2003, there was already some book chapters and books that were written about correlative microscopy. One that was published in '87 by Hayat is covering a lot of things that we are already using. We are now using as a routine. And I encourage you to search for this book if you have the chance. It's very interesting. But then since 2012, we've had a very significant increase in the amount of use of CLEM, the literature associated, and the technique which are corresponding. And this results in more than 81, in about 81 chapters of CLEM in this book series. And again, I would encourage you to have a look at those books. Miriam and I have been contributing a couple of chapters in those in this series. But as I said in the introduction, one of the part of correlative microscopy is in-resin fluorescence. That really came as a breakthrough in 2009, around 2009, 2011. And that was the first time we used it with fluorescence preservation of GFP. But before that, we already had some hints that we could preserve fluorescence or create fluorescence in resin blocks. And that could help into guiding us into volume for electron microscopy. So as I just said, there were two landmark papers that kickstarted in-resin fluorescence. The first one came from Susan Nixon in 2009, where she figured that she could preserve fluorescence in high pressure frozen free substituted in Lovicryl resin, and she could preserve the GFP and the RFP in the resin. And this was a brilliant proposal, and it was taken up marvelously by Wanda Kukulski and the whole group that she was working with, where she managed to keep the fluorescence in the sections and use that as a guiding to find out very fine structures such as endocytic vesicles at the surface of yeast. And that really kickstarted the field, and everyone started to look into unsectioned fluorescence preservation. So they rapidly got a paper on that and a book chapter, and we all took on this idea to develop our own approaches, and it became widely used. But we also figured quite quickly that it was not a simple technique, and it started to become a lot of courses for doing in-resin fluorescence because it's just not copy pasting a protocol from the book chapter. It requires a lot of things that cannot be taught by writing a book. And so what I would like to approach today is about the underlying mechanism, which are about preserving the protein for keeping them fluorescent in resin. So for that, I'll come back to some old publication by Kellenberger in 87 again, where he first mentioned in a very clear way the notion of hydration shell around proteins. And so what we see here is a representation of a protein. And as you can see, we have plus and minus charges, positive and negative charges at the periphery of the protein. And those interact with the positive and negative sides of water molecule, which is a polar molecule and polar solvent. And this hydration shell allows the protein to remain segregated from the surrounding proteins, but is also used as a scaffolding for the protein itself. I'll come back to that point in the next slide. What is important is at the macro scale, as we can see here, if you remove the full hydration shell, then the positive and negative charges of the protein will look for interacting with other positive and negative charges of other proteins around. And so this is where we will see aggregation patterns. If we look at proteins which are homogeneously separated, they'll aggregate in a completely random manner and it's unpredictable. Assuming you would have some supporting and scaffolding proteins around, then you'll have aggregation around these scaffolding proteins. So the larger proteins will remain unmoved and the smaller one will aggregate to it. And if you have a bunch of fibrillous structures, they will also aggregate in a completely unexpected and unpredictable manner. So that was how they presented it. And of course, they also introduced some images to support their arguments. And that's what we can see is that they looked at the plasma aggregation in cells and comparing glutaraldehyde and room temperature dehydration in ethanol, which is a very good technique, but has some artifacts which are nowadays well known, well established, but still it's good to remember. And when we read this paper, we see that the DNA is aggregating in a completely clumped manner, similar to what we see here. Then they compared it with PLT in ethanol. And this is also a very great technique, but it is not artifact free. And here is more reminiscent of the B situation we see here in this theoretical view. And then high pressure freezing and free substitution in acetone gives a much less condensed aggregation. It's maybe more remnant of the A situation, but nowadays, when we compare high pressure frozen free substitution sample with cryo data, this is considered as being the closest to native we can get aside from cryo imaging. So this is at the macro scale, but we can also look at it into the nanoscale. And basically the protein itself is not only surrounded by protein, but also embedded into the solvent, to the water solvent, which helps as a scaffolding and supports the protein from collapsing on itself. And here at GFP, if you consider it as a barrel, if you remove the water, it will just simply collapse and not be able to resonate anymore. And one of the point of the GFP is that this barrel allows to resonate because the protein inside can be freely moving with this protective layer around. And so when we think about electron microscopy, there is 5 steps that we cannot avoid in biology. The first thing is we need to immobilize the biological material. Then we will need to dehydrate, enhance the contrast, embed the sample, and thin it down so that we can see it with the electron microscope. But each of these steps is associated to artifact and problems. The first thing is when we do the immobilization, we can do chemical fixation. But chemical fixation may cause protein aggregation, we've just seen that, and protein quenching, because basically if you create a chemical bond within a protein, it cannot resonate anymore. If you do a physical immobilization, like vitrification by plunging or high pressure freezing, you might have some denaturation by dehydration. And I'll come back to that point in a minute, but basically when the water crystallizes, it causes a phase segregation that will dehydrate your protein. Then the dehydration, as we just mentioned, it can cause protein denaturation. The contrast enhancement, we're adding some chemical fixatives and heavy metal salt to the protein so we can impair the resonance of the proteins. Then the embedding, depending on the resin, we might have some interaction between the resin and the protein, causing, again, some quenching. And last but not least, the thinning, which means that we physically reduce the amount of fluorescent proteins that we can see into our lamella, meaning that we have harder time to see the fluorescence. So let's go to all of these points one by one. Immobilization. we have the chemical fixation, and the chemical fixation, whether it's glutaraldehyde or formaldehyde, causes bondings between the protein or within the protein. And this is not artifact free, but it has a lot of advantages, including the price. It's very cost effective, can be done at the corner of the bench. It doesn't need special equipment, but essentially it will damage or reduce your fluorescence. If you do cryoimmobilization, we're talking to something which is nearly instantaneous. It's happening within about 10 milliseconds. So this prevents some fluid exchanges, and it's usually tolerant to, it's not impacting fluid transfers within the cell. And it's been, as we said already before, considered as the closest to native we could get in plastic EM. But they are not artifact free. Either situation. The chemical fixation, you have to face diffusion issues. And the further you go inside the sample, the longer it takes for the chemicals to diffuse through the first layers, which have been fixed, to reach the center. And then you'll have a gradient of quality between the center of your sample and the periphery. And the fixation is not going to be homogeneous across the whole thing. But high pressure freezing, on the other hand, is not artifact free either, because if you have a bad gradient of temperature, then you'll end up having crystallization, massively bad crystallization, up to potentially acceptable vitrification. But we would not recommend looking at structures which are near massive ice damages anyway. So to go back into more details of this dehydration that is caused by the crystallization, if you consider the protein being scaffolded by water, when the water crystallizes, a crystal wants to be pure. So what will happen is that the water molecule will extract from the proteins to create a pure crystal of water. And by doing so, it will progressively capture the water molecules which are scaffolding the protein and leave the protein dehydrating on itself. And so this is what is very genuinely presented here. Basically, the water is scaffolded by water molecules surrounded by liquid water. And then progressively, the water scaffolding will escape, start to crystallize at the periphery more and more until you have a completely dehydrated protein surrounded by large ice crystals. So if we think about that with real images, here it would be a Semovis image, so vitrified material and image in cryo conditions. Then we can substitute the water content and get a perfect plastic ultrastructure where you have no major damages. Then you will start to see minor ice damages where basically you have some extraction and you have some segregation with ice nucleus close to proteins. And then worst case scenario, you have a massive dehydration. We call that ice bombs. And basically you have water on one side and aggregated protein on the other side. Now the dehydration. When we do dehydration, it's a bit different from substitution. The dehydration is a stepwise process where we will progressively remove the water and exchange it with a solvent, ethanol in most of the cases. And this occurs in steps, and this causes a phenomenon of fluid exchanges. The water has to move away and is replaced progressively by the ethanol. And this is basically, we are diluting the water content of the cell. These movements are accompanied with some washing steps, and basically we're causing some extraction of the protein structures. When we do free substitution, it's a bit different because everything occurs at minus 90 degrees Celsius. And at this temperature, the water present in the cell is solid. And so instead of having movement of liquid water, we have solid ice that is progressively diluted piece by piece by the acetone that replaces it. And this is why we talk about substitution rather than dehydration. But the two phenomena aims at removing the water content of the protein. The second version is much smoother and milder to the sample. So again, the idea is to be able to remove the water while preserving somehow the hydration shell. And this is why a lot of protocols nowadays are preserving the hydration shell by not dehydrating completely the sample, but by keeping 5% of water inside the structure, meaning that the proteins will remain structured because all the scaffolding water molecule will not be extracted during the dehydration. But what is surrounding the protein will be substituted by the solvent, whether it's acetone or ethanol. So the idea is to have the water molecule inside the protein remaining and the water molecule outside the protein being exchanged by the solvent. Then the contrasting. The contrasting is something very, very important because our proteins are made of CHO, and our resins are made of CHO, carbon, oxygen, hydrogen. And so basically, there will be no difference in mass, and so no difference in contrast under the electron microscopes. So that's why we add some contrast by bringing some heavy metal salt that will interact with the proteins, go inside the cavities of the protein, and remain here so that when the electrons are hitting the heavy metal, they will be stopped or diffracted, causing a differential image on your electron microscope screen. And essentially, we're using most likely we use two heavy metal salt. One is the uranyl acetate, which is approximately 20 times larger than a water molecule, and it cross-reacts with biological material and causes some autofluorescence. So the use of massive amount of uranyl acetate will be killing your fluorescence. The other contrasting agent we're using is osmium tetroxide, which is a highly reactive molecule composed of 4 oxygens that are reacting with everything they can around, and essentially binds to lipids, reacts with alkenes, and unfortunately darkens all biological material and turns it in pitch black, so preventing even bright light to go through your sample. But we need those elements. So the idea is, where do we find the balance between being able to see the fluorescence that we had natively expressed in the sample and having contrast in the electron microscopy? And the balance between those two is often very challenging and tends to be towards a very minimal amount of heavy metal salt to not quench the fluorescence. Then we still have the embedding. The embedding means we have to remove the water from the molecule from the sample, replace it by a solvent coming with fixatives and contrasting agent, and then we have to replace that by a resin. And this resin has a very long polymer chain. As I said, it's also composed of carbon, oxygen, and hydrogen, and they are not completely compatible with protein nature. So they will mesh, and they can be of two types. Part of the protein of the resins are cross-reacting with amine groups of the proteins, like epoxy resin, meaning that they cause a very great embedding quality and interaction with the proteins. However, by interacting with the protein, they block it, and that's why EPON is quenching the GFP. On the other hand, we have methacrylate resins like HM20, LR white, or R221 resin, and those ones are reacting among themselves, not interacting with the protein. So basically, instead of cross-linking with the protein, they surround the protein, creating their own mesh, giving still freedom for the protein to resonate and continue being fluorescent. However, this infiltration occurs also in gradients, so we also have fluid exchanges, and we might have some interaction, so that might cause also extraction and deformation in the ultrastructure. But technically, since we've discovered, Susan Nixon discovered that the methacrylate resins like HM20 could preserve the fluorescence, we've all been going into that direction, aiming for methacrylate resins to keep the fluorescence. There are some publications around which are illustrating the use of epoxy resins for preserving the fluorescence, but they are still not the majority of the studies we see around. And with that, I'll hand over to you.
Yes, thank you. So, well, as Xavier already mentioned, we wrote an extensive review on the topic, and I don't want to go through all the details of this again. You can look it up. But I wanted to highlight, we do have a large list of fluorophores that have been used for in-resin fluorescence, although, as you see on the right side, the genetically encoded labels that you can use, like GFP, RFP, there's only a few of them, and there is lots of publications. As you see, they all tried to make in-resin fluorescence possible. What I want to point out today is more looking at this side of the list, which is even longer, and you may see that most of this has come from the lab that I've been working in, and I'm still working on these things. We used non-specific stains, and these actually also have their pros and cons. Of course, we want to have specific labels, and I fully understand the need to use GFP, RFP, and all those labels to have specific structures stand out. But I want to point out there is also other options for correlative microscopy. that can be very valuable. As you see here, we have done lots of this unspecific staining, and the fluorescence can be very well preserved in both epoxy resins and in HM20 we have used mainly. My favorite stain basically is this one here, which is even compatible with osmium. If you remove it from the free substitution at something like minus 50 degrees, you still get very nice fluorescence, and this is DII or DIO stain together with Nile blue. You get very nice description of cells. For example, this is a monolayer of cell, and this works for single cells, this works for tissues. It does not give you, as I said, it does not give you very specific staining, but it really nicely describes the sample on a histological level, which is very helpful in many cases to find your structure of interest and guide you when it comes to just looking for specific areas, especially with skin samples as we have here on the right. This project was really aiming to find sweat glands in human skin, and they have a very distinct morphology. It's easy to distinguish them when you see them by structure, but the question was really like, where in our resin block do we have this structure? If we want to cut through the entire sweat gland, we don't want to lose half of it by just trying to find it by serial sectioning TM. Or looking at the serial section, that's a lot of work if you do it with TEM. And this was really making us much more efficient to find our structures. Sorry. This is another example of work with human skin that we did. This goes back to my PhD thesis, where we were looking at pigmentation in human skin. And we also figured out that just using this histological in-resin fluorescence to describe the skin biopsy by using the confocal on the resin block, we could actually see a reflection signal coming from the pigmentation as we see it here. This is really just pure reflection recorded in the confocal. And if we zoom in a little bit, we see the overlay here or the combination, and you see how nicely this actually corresponds. So this was very helpful already, trying to figure out if we have a sample, for example, with lots of pigment, or if it's rather less, and that helped us to distinguish what we wanted to investigate in closer detail. This is again an example of human skin done a few years later, just to highlight that you can not only use this in-resin fluorescence in sections, they still fluoresce. If you, this is basically going back to the idea of poor man's confocal. You just do serial sectioning and record it by a standard light microscope, a wide field, and you still have very wonderful resolution also on the light microscopic level. But you can also stain those sections additionally. So here we have the combination of DARPy to get this to get the nuclei, and here we have even toluene in blue, which is not colored. Here we have it in a colored representation. So all of this is possible with these samples. And this is just to show the same area. Again, this was now recorded with an SEM, and you see it's tiling. And here's the details, and you see how nicely, for example, the collagen is preserved in these high pressure frozen samples. And again, finding the sweat gland structure here. Now, what also was very important for us to figure out at some point where exactly we lose the fluorescence. We have heard all the theory by Xavier earlier on, and now this is how it looks basically in images. This was done with a microfluidic device on a light microscope, where we could exchange the medium directly on the light microscope and basically watch at which step we actually lose the fluorescence. So here we have a GFP signal and a BFP signal in the same cell line. This is still live, and then we start the fixation. And even if we have a high concentration of glutaraldehyde, which is said to have an autofluorescence, it didn't disturb us too much. We still have nice fluorescence. And you may see that this is different areas of cell culture for every single image that we took, because we didn't want to have any quenching effects if we look at the same region of interest again and again. So different cells every time. Now we have uranyl acetate staining, still very nice fluorescence, everything very nice. We continue with 1% osmium tetroxide, and we still have the fluorescence. Both BFP and GFP is still very nicely alive and no change in signal. Then we start dehydration, 25% ethanol, 50% ethanol. That looks slightly different, but I think this is just an artifact here. The green fluorescence is really still nice. And we're going into 75% ethanol, 90%, 98% ethanol, and 100. And this is where we actually start to lose the fluorescence. So osmium and uranyl acetate did not impair the fluorescence. It was the full dehydration, basically, which goes back, and this was done at room temperature. But this goes back to what Wanda Kukulski and Susan Nixon also said. If you don't fully dehydrate the sample during free substitution and keep a little bit of water, you will maintain the fluorescence. And this is now the epoxy resin, and you see there we have all the fluorescence of the epoxy. And yes, it doesn't get nicer. This is to highlight the actual influence of uranyl acetate. Just comparison, same cells, this is still live, and now we have the polymerized samples. And as you see, we do have some fluorescence left, even in epoxy resin. This is now done without osmium, but we do get the same fluorescence signal, basically, with all kinds of different excitations. And only this part is really specific. This is the cell, and all the rest is background. So we should not underestimate the influence of autofluorescence of uranyl acetate, which, on the other hand, can also be used. So we have also had studies in the past where we actually used just uranyl acetate fluorescence from the resin-embedded sample to guide us, instead of adding fluorescence by stains.
Miriam, you have 2 more minutes.
Okay, we'll speed things up. I'll just run through this. Same thing again, same experiment again, but a far red stain. And this is something that I would like to recommend to everyone. If you want to do in-resin fluorescence, the far red stains are actually so much more stable. I know GFP and BFP and RFP are around, and they are all established, but try to go for far red stains. They are so much more stable, and you get wonderful fluorescence already, also with epoxy embedding. And this is a FIBSAM of this. Now, Xavier already mentioned the R221 resin, which was specifically designed by CryoCapsule to maintain fluorescence in these resin samples. And this is my very first experience with R221. And you see it's not perfectly preserved here. We have the cell here. High pressure freezing didn't go well that day, but still. You see we have some fluorescent spots in the cell here. We find it again in the TM section, and it was about these vesicles where we wanted to find those nanoparticles, or those nanocompartments back. So this worked. This was really, as I said, my first trial ever with R221, and it worked on the first trial. So that was really good experience. So this is what I would recommend to go into. And you see, it also gives us a nice ultrastructure. This is a Fib cross-section. Even with this not perfect freezing, we do get to see all kinds of details with a very low amount of uranyl acetate contrasting only. And this is something that we have to highlight. You don't need so much uranyl acetate here, and you don't need osmium at all to get good contrast.